For a long time, seminal plasma was described as little more than a transport medium, a simple fluid that carried, fed, and protected sperm cells on their journey. But modern reproductive science tells a very different story. Seminal plasma is not a passive vehicle. It is a biologically active signaling fluid, one that actively prepares the female reproductive tract to support conception, implantation, and even the earliest stages of embryo development. Let's start with what it actually is. Seminal plasma is the fluid part of semen, making up around 95% of the ejaculate. It's assembled from several glands. The seminal vesicles contribute the largest share, roughly 2/3. The prostate adds about 1/4, and smaller amounts come from the epididymis, the testes, and the bulbourethral glands. Inside this fluid is far more than just sugars and salts. Yes, it contains fructose for energy, zinc and citrate. It contains the proteins that let semen coagulate and then liquefy, but it also carries a remarkable cargo of signaling molecules. Chief among them is a molecule called TGF-beta, or transforming growth factor beta. Its concentration in seminal plasma is extraordinarily high, far greater than in blood, in colostrum, or in breast milk. Alongside it are prostaglandins and a whole orchestra of cytokines, some that promote inflammation and some that calm it. And there are tiny packages called extracellular vesicles, carrying proteins, DNA, and RNA that can talk directly to the cells of the womb. So what does all this signaling actually do? First, it builds immune tolerance. An embryo carries genetic material from the father, so to the mother's immune system, it looks partly foreign. Seminal plasma helps solve this. It delivers paternal signals together with TGF-beta and prostaglandins, encouraging the growth of regulatory T cells. These cells teach the immune system to tolerate rather than attack the developing pregnancy. Second, it triggers a controlled remodeling of the womb lining. Seminal plasma sets off a carefully managed inflammatory response, recruiting immune cells that reshape the tissue and improve the receptivity of the endometrium, making it more welcoming to an embryo.
Third, it supports the early embryo directly. The cytokines released, such as GM-CSF and leukemia inhibitory factor, have nourishing effects on the embryo and can promote the growth of new blood vessels at the site where the pregnancy takes hold. And there's a fourth role worth mentioning. Because seminal plasma reflects the health of the glands that make it, it can act as a diagnostic window. Low fructose can point to a problem with the seminal vesicles. And PSA, the marker used today for prostate health, was first discovered in semen. But here's the honest, important question for fertility care. Does all of this rich biology actually translate into better success rates in IVF? The answer, so far, is genuinely mixed. A major Cochrane review found that applying seminal plasma around the time of treatment may increase clinical pregnancies, but showed little or no difference in live birth rates, and the quality of that evidence was rated low. Several well-designed trials have found no benefit at all. Why the inconsistency? The concentration of these active molecules varies enormously from one man to another, and even between different fractions of a single sample. The effect seems to depend on the full, undiluted cocktail rather than any single ingredient. And there's a theoretical concern that prostaglandins could trigger unwanted contractions of the womb. So where does that leave us? The science is clear on one point. Seminal plasma is absolutely not merely a transport medium. It is an active immune-modulating and embryo-supporting signaling system that helps prime the body for pregnancy. But when it comes to using it as an add-on treatment to improve live birth rates in IVF, the evidence simply isn't there yet. For now, it remains a fascinating area of research rather than a proven part of standard care. Understanding this distinction between compelling biology and proven clinical benefit is exactly what good, evidence-based fertility care is all about.